In the clinical standpoint, we find that this issue of resistance, you know, either as a really big problem, something that we really need to be worried about, or yes, it's going to be a problem, but that's not for something that it's over the horizon. It's not here quite yet. Urgent threats, serious threats, concerning threats. Urgent threats right now are going to be things like this multi-drug resistant gonorrhea. Yes, an STD that is now resistant to almost all antibiotics we have. Yeah, how scary is that? Everyone's thinking, oh, it's gonorrhea, it's not that big a deal, blah, blah. No, gonorrhea can be a pretty bad thing. It can ascend, for guys, it can ascend up the urethra, do damage to the prostate, keep ascending, do damage the epididymis, the testes. Now it's either direct damage due to the gonorrhea itself. What we find, though, is most of the damage is going to be due to the inflammatory response because of the presence of gonorrhea. Same thing with women. Gonorrhea is going to ascend up the vagina, pass through the cervix into the uterus, continue ascending transversely through the fallopian tubes to the ovaries. Presence of the bacteria does some minimal damage. The over response from the immune system, the inflammatory response, is going to cause extreme damage, rearranging of the architecture of the cervix, the fallopian tubes, which will not allow, which in extreme cases, pelvic inflammatory disease due to gonorrhea, will interfere with passage of eggs through the fallopian tubes, basically rendering the women sterile. Urgent threat. Serious threats, ones that we can still deal with, but it's becoming quite an issue. MRSA, methicillin-resistant Staphylococcus aureus. It's resistant to a lot of the drugs. Not all, a lot. We still have vancomycin and a few other drugs. Can't think what other drugs there are right now. So MRSA spreads through the hospitals. Hospitals don't like to admit it, but they're a pretty big source for these infections. What we find happens, since they're resistant to most other drugs except methicillin, or most other drugs, specifically methicillin, which is what we use for Staph aureus. Vancomycin's the last magic bullet, but we're running into VRSA, vancomycin-resistant Staph aureus. Once that starts to spread heavily, then this is going to go from an serious to an urgent. Concerning threats, these are many others I'm not going to go into right now. New approaches. This is going to be in medical and clinical research right now, as they're in drug research, as they're trying to come up with new ways. They're always trying to come up with new drugs. Problem is they're running out of ideas. They're hitting the wall on what can be designed. Every time they try to design a new drug, turns out that the resistance issues that already exist are going to allow for resistance to the new drugs. So what they're looking at are other approaches that are not antibiotic-based. RNA interference, mess with, use small RNA fragments that get in and will bind to bacterial messenger RNA. Our cells have enzymes that find double-stranded RNA and cut it because it's thought of as being viral. Single-strand RNA, messenger RNA, tRNA, it ignores. Double-strand cuts. So come up with these short RNA strands. These small silencing RNA fragments, can get into the nucleus, interfere with gene expression. They can get into the bacterial cells, interfere with gene expression there. CRISPR, the two women that independently from each other isolated, characterized CRISPR. Turns out here, in year of our Lord 2020, they just were awarded the Nobel Prize for this. CRISPR is a bacterial cell defense mechanism. It's a proteinaceous RNA molecule that goes and surgically cuts DNA fragments, RNA fragments that are viral based. It does not recognize, does not interact with bacterial genome DNA, but it goes and cuts bacteriophage DNA. Awesome. We're now looking at non-cultivatable bacteria. Turns out we haven't isolated and identified and been able to cultivate the majority of the bacteria out there, majority of the microbes. So what they're trying to come up with is a way to isolate these bacteria. May not be able to culture them in the lab, but isolate and identify whatever compounds are present. And then we're also looking at bacteriophages themselves. If we can tailor a bacteriophage, a virus that can only infect bacteria, tailor it in such a way, and then come up with a delivery mechanism. One goes in, hundreds, thousands come out, hundreds, if not thousands, come out, spread to other bacteria, and in no time, no way can it infect human cells.